afternoon. So today we are looking at glycolysis and Krebs cycle. So let's define cellular respiration. So this is where energy flows into an ecosystem with sunlight and leaves and heat. Living cells require energy from outside sources to do work. The work of the cell includes assembling polymers, membrane transport, moving and reproducing. The chemical essentials which are essential to life are recycled. Photosynthesis generates oxygen and organic molecules which are used in cellular respiration. Cells use chemical energy stored in organic molecules to generate adenosine triphosphate, which powers work. So cellular respiration includes both aerobic and anaerobic respiration, but is often used to refer to aerobic respiration. Although carbohydrates, fats, and proteins are all consumed as fuel, it is helpful to trace cellular respiration to sugar glucose, as you can see in the equation below. The transfer of electrons during chemical reactions releases the energy stored in organic molecules. The released energy is ultimately used to synthesize an adenosine triphosphate. In cellular respiration, glucose and other organic molecules are broken down in a series of sets. Electrons from organic compounds are usually first transferred to NAD+, which is a coenzyme. As an electron acceptor, NAD+ functions as an oxidizing agent during cellular respiration. Each NADH+, which is a reduced form of NAD+, represents stored energy that is tapped to synthesize adenosine triphosphate. NADH passes electrons to the electron transport chain. Unlike an uncontrolled reaction, the electron transport chain passes electrons in a series of steps involved instead of one explosive reaction. Oxygen pulls electrons down the chain in an energy yielding tumble. The energy yielded is used to regenerate ATP. So, within cellular respiration, harvesting of energy from glucose has three stages: glycolysis, citric acid cycle or Krebs cycle, and oxidative phosphorylation. Glycolysis breaks down glucose into two molecules of pyruvate. The citric acid Krebs cycle completes the breakdown of glucose, and oxidative phosphorylation accounts for most of the ATP synthesis. So the process that generates 90% of ATP is called oxidative phosphorylation because it is powered by redox reactions. So that's reduction and oxidation reactions. A smaller amount of adenosine triphosphate is formed in glycolysis and a citric acid cycle by substrate level phosphorylation. For each molecule of glucose degraded to carbon dioxide and water by respiration, the cell makes up to 32 molecules of ATP. When glucose enters a cell from the bloodstream, it is immediately converted to glucose 6-phosphate. Once this phosphate is formed, glucose is trapped within the cell because phosphorylated molecules cannot cross the cell membrane. Glucose 6-phosphate can then be converted to pyruvate. Glycolysis is defined as a series of 10 enzyme catalyzed reactions that break down glucose molecules. The net result of glycolysis is the production of 2 pyruvate molecules, 2 ATPs, and 2 NADHs/H pluses. Glycolysis, which is essentially sugar splitting, breaks down glucose into 2 molecules of pyruvate. This occurs in the cytoplasm as 2 major phases: energy investment phase and energy payoff phase. Glycolysis occurs whether oxygen is present or not. So let's look at mitochondria now. So mitochondria are energy converting organelles in the cell, also known as the powerhouses of the cell. The term mitochondrion is derived from a Greek word mitos, which means thread, and chondrion, which means granule. They are inherited maternally in the cytoplasm of the egg. They contain their own DNA, which is circular, so can be described as semi-autonomous organelles. They have a double membrane structure, an outer mitochondrial membrane and an inner mitochondrial membrane, which consists of highly invaginated cristae that contains proteins for oxidative phosphorylation. The mitochondrial matrix is the site of tricarboxylic acid cycle, also known as Krebs cycle, and contains mitochondrial DNA. So the conversion of glucose to pyruvate in glycolysis is a key metabolic pathway in most living things. So in aerobic respiration, in the presence of oxygen aerobic conditions, pyruvate is converted to acetyl-S-CoA. Pyruvate diffuses across the outer mitochondrial membrane and then is carried by a transporter protein across the inner mitochondrial membrane. Once inside, pyruvate dehydrogenase complex catalyzes the conversion of pyruvate to acetyl-S-CoA. So the oxidation of pyruvate to acetyl-CoA is a step that is carried out by a multi-enzyme complex that catalyzes 3 reactions: oxidation of pyruvate and release of carbon dioxide, reduction of NAD+ to NADH, and combination of the remaining 2 carbon fragment and coenzyme to form acetyl-CoA. So looking at the tricarboxylic acid cycle, Krebs cycle, or citric acid cycle, these complete the breakdown of pyruvate to carbon dioxide. The cycle oxidizes organic fuel derived from pyruvate. The citric acid cycle has 8 steps, each catalyzed by a specific enzyme. The acetyl group of acetyl-CoA joins the cycle by combining with oxaloacetate, forming citrate. The next 7 steps decompose the citrate back to oxaloacetate, making the process a cycle. The NADH and FADH2 plus produced by the cycle relay electrons extracted from food to the electron transport chain. The starting molecule oxaloacetate binds with acetyl-CoA to make citrate. This is progressively transformed throughout the cycle. It always turns back to oxaloacetate to start the cycle again. The energy released is stored in the reduction of free NAD plus to free NADH, 1 FAD to FADH, and the synthesis of 1 GTP, which is converted to 1 ATP.